A very good morning, students. Now, this is a lecture on the mutation, which is a very important topic usually asked as a short note in the examination. Now, before we learn to the mutation, I will tell you that the characters or traits or abnormalities, they are transmitted from one generation to next. And the characters which are transmitted from one generation to the next generation, okay, they are called as heritable traits or heritable structure, say characters. And who is responsible for this inheritance, okay, why they are transmitted from one generation to next. We have already learned that there are presence of chromosome in the nucleus, and at the time of the fertilization the ovum and the sperms, they ultimately transmit these chromosomes to the next offsprings, okay, to the offspring. So along with the chromosome, these characters are transmitted, okay. So one of the inheritable structure is the chromosomes, okay. And then if we go still on the finer aspect, what does a chromosome contain is there are lots of genes, about 30,000 genes which are sitting on the chromosome. They are actually responsible for transmission of the characters. So in this chapter we will learn that what happens if these inheritable structures, that is chromosomes, or that of the genes are uh, becomes abnormal or altered, okay. And then what will be the effect into the I will say in transmission of the characters. Since we have learned the chromosomes, okay, both the numerical where the chromosomes were more in number or less in number already, okay, we will limit ourselves in this lecture to that of the genetic mutation, or the mutation or abnormal gene transmission from one generation to next, or within the same somatic mutations, okay, that we will learn. So I am passing to the next slide. Hello, let's come to the actual definition of the mutation. The heritable changes in the genetic material of an individual is called as mutation. What is heritable changes? Heritable changes means when after the mutation, whatsoever changes has taken place in gene or chromosome, they can be transmitted to the next generation, or in case of somatic cell, they can be transmitted to the next cell division. Okay, so that is called as the heritable changes in the genetic material. And in the last slide I have explained what is genetic material. Either it is chromosome or the genes sitting onto the chromosomes which may show the to say this mutation, okay, mutation. So this is the definition of the mutation. Once again I will repeat, it is a heritable changes in the genetic material of an individual, which is called as the mutation, right. The mutation or these heritable changes may occur in a single gene or in chromosome, as I have already said you, okay. If heritable changes occurs in the structure of a gene, okay, and gene is what? It is nothing that a part of the DNA molecules, okay, double stranded helical structure of DNA, and part of it, okay, will be a structural gene, okay, as you must have learned in your biochemistry classes, okay, the definition of gene, okay. So if this heritable changes occurs in the structure of a gene, then this is called as genetic mutation or point mutation. As we will see why it is called point, because we will see where ultimately in a gene this change may occur at one particular point at one nucleotide base pair. When these changes occurs in chromosome, it is called as chromosomal mutation. Thus, mutations are of two different types. The point mutation, that means mutation within a gene, or it is because of the changes in chromosome, which may be either numerical changes, means the chromosome may be more, okay, Okay, trisomy or chromosome may be less. For example, we learn about monosomy, or it may be the change in the chromosomal structure. Say for example, deletion, isochromosome, ring chromosomes, okay, translocation, like that. Okay, these kind of the mutation are called as the chromosomal. But in this lecture, we are not going to talk about the chromosomal mutation. We have already discussed the chromosomal mutation at great length in the last few lectures. Okay, here we will only concentrate on to the gene mutation or the point mutation. Now, because of the mutation, there occurs the genetic variations. That means when the mutation takes place, then the variation will occur because something new will produce because of the mutation. Either it is a beneficial effect will be there, or it may be a I mean to say harmful effect will be there of the, mostly it is harmful, but sometimes the beneficial effect of the gene mutation also occurs. And this is constantly going on for millions of years. Okay, mutation, it is not that in one person the mutation has taken place and then its effect will be seen. Okay, the beneficial or bad effect will be seen gradually, okay, extending into many years. Okay, many. Now this mutation, which is for the betterment or for the worst, um, for a individual, that is for a animal or the plant also, this is the basis of the evolution. Okay, what the Darwin has said, that it is the survival of fittest. How it is the survival of fittest? That means because of the mutation, which is a gradual process, an animal or plant develop itself so that he can survive in the nature. Otherwise he will be not be able to survive, he will die. Okay, and his species will no longer will be there in the nature. So this is how ultimately the mutation forms the basis of the evolution, and whatsoever evolution has taken from unicellular animal to that of the human being. Okay, now this is because of the evolution which has taken place into the genes. Okay, so new species are formed because of this evolution. Okay, now I am moving to the next slide. Now there are two types of mutations. Okay, we are now limiting only to the gene mutation, not talking of the chromosomal mutation. So just think that we are talking of the gene mutation, the mutation or abnormal or inheritable change which takes place within the gene, that was also called as the point mutation. So we are limiting ourselves to the gene mutation now, because chromosomal, as I said, we have already uh, seen. Okay, now there are two types of the gene mutation: somatic and germinal mutation. It is somatic mutation or germinal mutation. Let us see what is somatic and what is germinal mutation and what is difference between the two. Okay, mutation occurring in somatic cells, since we are made up of billions of the somatic cells, okay, which have formed our body. I mean say all those cells which are present in our body except the, that is egg and the sperm, that is ovum. Okay, and the sperms which are they are not the somatic cell, they are the germline cells. Okay, or germinal cells or gametes, they are called as gametes because they are different, because they contain the haploid number of chromosomes. Okay, their genetic constitution is different absolutely than the somatic cells. Okay, when it occurs in germ cell, it is called germinal mutation, and when it occurs in somatic cell, that is called as the somatic. So you should be clear in your mind that the billions of cells in which of which our body is made up of, they are somatic cells, and those gametes, okay, sperm and ovum, they are uh, the germ cells. Okay, they are the germ cells. So if change or mutation is taking place in germ cell, that is germline mutation, and if it is the gene changes are taking place in the somatic cell, it is somatic cell mutation. Okay, I have again repeated it, okay, just for your benefit. Now coming to the difference between the somatic mutation and the germinal mutation. Okay, the somatic mutation occurs in somatic cell, as I told you, and therefore it is not inherited, okay? Because your somatic cells, say the cells of the epithelial lining of the gastrointestinal tract, which are very rapidly dividing, if something goes wrong in those cells which are dividing in the epithelial lining of the small intestine, it is not going to be inherited. It will not go to your next generation, okay? Because these are somatic cell. But if this occurs in a germ cell, that is in ovum or sperm, hence it is inheritable. It will be this mutation or this change in the gene will be transmitted to the next generation, okay? It will be, but the somatic mutation will not transmit. It will be limited in that organ only. It will remain, whether it is beneficial or it is harmful, it will be there only. So it will not be transmitted to the offspring, okay? Now, second difference is that it produces local phenotypic changes in individuals. Somatic mutation will, if it is harmful even, then it will just have a very local effect, okay, where the cells are dividing and producing those mutate gene, okay, where the cells are with those mutate gene or abnormal gene. That abnormal gene will have abnormal product in the form of protein, enzymes, hormone, whatsoever it is there, and it is in that particular area only its effect will be seen, not even in the whole body it will be seen, okay? But in case of germinal mutation, since it is the mutation in the gene which was in the, I mean say, in the sperm or the ovum, okay, it will so after fertilization a new individual will develop, and it's all the cells, all the billions of cells and the body of that newborn child will be having effect everywhere. It will have thus a generalized effect in us, because each and every cell is having that effect. On other hand, the somatic mutation was having effect only where in that organ where this has occurred, but the others organs or other parts of the body, okay, they are normal, they are having the normal somatic cell, right? Now, the third difference between somatic mutation and germline mutation is that individuals are mosaic. Mosaic, I have told you when I have taught you the mosaicism and chimerism, okay? Mosaic is an individual which shows the two cell lines, okay? Two different cell lines. Thus, the it will have most of the cell in the body will have the normal somatic cell, but where this mutation has occurred, that will those cells will be also the mutate cells will be there. Thus there will be two different cell line, one normal cell with the normal genes on the chromosomes, and where mutation has occurred with the mutate gene on the cells within the somatic cell. But here in the germinal mutation, individual is not a mosaic, because all his cell, 100% of his cell, okay, somatic cell, okay, they will carry this mutation, because this was transmitted through sperm or the ovum, okay, which has formed a single fertilized egg, which was containing this mutate gene, and every cell division following that will have the mutation. Next slide. Now, the mutation which is occurring in a gene, it may be always not a harmful, will have always not a harmful effect, because of that abnormal changes which has taken place in gene. It may be sometime beneficial for a person also, okay, or for a plant or for an animal, it may be the beneficial. Thus the mutations are also of two different kinds: gain of function mutation, where the function is has increased, okay, or the loss of function. Most of them are mutation which takes place, they are for the loss of the function, because a already existing gene will show some abnormality which is more towards the loss of the function mutation. So what is the loss of function mutation? Okay, they are very common. Okay, this results in complete inactivation of gene. When this mutation will take place, either the whole gene will get inactivated. Okay, so that means whatsoever hormone, protein which it was producing, or enzyme which it was producing will not be produced, or if it will produce, it will be produced with the reduced activity or with the altered activity, will have the different effect, okay, than the normal. Okay, we will see the example later on. This mutation is also called as the knockout or the null mutation. Okay, that is the reason why, because loss of function is there, that's why it is called as the knockout or the null mutation. Most of the loss of function mutations are recessive, that you should also keep in mind that they are recessive in nature. Now we come to the second type of mutation, okay, which is the gain of function mutation, okay, where new functions are acquired after the mutation or change in the gene. This result in overexpression of gene. Gene is producing more, okay, gene products are overproduced, okay, and that is also harmful sometimes, okay, it is also. So that this kind of mutations are mostly dominant, and most of the gain of function mutation are responsible for cancer production. So when because of the gene mutation, when the product of the gene are forming and they are overproduced, okay, so this ultimately will lead to the cancer also in some situation. I'm not explaining how, but you just remember that there are two types of the mutation: loss of function or gain of function. Gain of function may be sometimes beneficial, but still it is not beneficial, as it can produce the cancer in some cases, okay, in some cases it is beneficial. Mostly the loss of function is always not beneficial, and which is a common type of the mutation. Okay, moving to the next slide. Now, what is the molecular basis where this mutation, where this change is actually taking place within the gene, that we will now learn. Okay, first we have learned what are the different types of mutation in a gene. Now we will learn what are the molecular basis of gene mutation or the point mutation in the gene mutation. We are on the gene. Okay, mostly during replication and transcription, okay, of the DNA. What is replication and transcription? Transcription is usually which is from the DNA molecules, which is a double-stranded DNA molecule which separates, and against the single strand of DNA, a new strand is formed where message is sent, okay. Hmm, and this message goes in the form of messenger RNA outside. That is transcription. Replication means where during the mitotic cell division or meiotic cell division, when new DNA has to form at that time. These are the two important aspects, okay, one in germinal mutation and another in the somatic mutation, where these are the two occasions where something may go wrong in the gene when they are forming new, okay, when the DNA molecule is forming new, okay, against what is existing, and another existing DNA molecule will give rise to a new molecule, okay. So this is the time where ultimately the abnormality can take place, okay. So there may occur the addition, deletion, or substitution of a single nucleotide pair in the DNA molecule. What is a nucleotide pair? Single nucleotide pair. This is the basic unit of the DNA molecules, okay? And so many nucleotide pairs, nucleotides, they just will join end to end and will form one strand, and against which and another strand will form, okay. So that way the DNA molecules will form double strand of the DNA molecule. And in a nucleotide, what is there? There is a nitrogenous base, there is a sugar molecule, and then there is a phosphate. That is called as a nucleotide. Remember your class in the biochemistry where they must have taught you about the molecular structure of DNA. Okay. Now at that time where this replication or transcription of DNA is taking place, this nucleotide pair may go wrong. Okay, pairing of nucleotide may go wrong, and we will just see how it may go wrong. Okay, so thus therefore the gene mutations are of two types. As when we see at molecular level, the gene mutations are of two types. That is, one type is called as substitution mutation, where a new nucleotide will come, a pair of the nucleotide will come in a DNA double helical structure of molecular structure of DNA. A pair of nucleotide will be coming here, okay, and it will substitute for in the already which are arranged in a line. Okay, this nucleotide pair, one may come and gain entry, a new one can come, or one may be deleted. Okay, so one may be deleted. Okay, that means a new nucleotide base pair, okay, I mean say whole molecule can come, okay, or it may be deleted. This is called a substitution mutation. I will tell you the example and will show you a diagram how this substitution mutation takes place in a molecule. Okay, then second kind of the gene mutation is the frame shift mutation. Okay, this is due to the deletion or insertion of a nucleotide base pair. Okay, so a new thing is deleted, I mean say added, that is insertion mutation, or a new thing is deleted. Okay, and therefore the frame will shift, and I will tell you how. Okay, when I will explain you, you will understand it. Let us first come to the first type of gene mutation, what is called as substitution mutation. Okay, substitute. In this one nitrogenous base, okay, of the triplet code is replaced by another base. Okay, so what is a triplet code? And you must have learned that triplet code is formed by the three nitrogenous base, okay, which are arranged in sequence. Three nitrogenous base, they will code for one amino acid. Okay, so there is a different codes for different amino acid. Okay, and this is called as triplet code. In this triplet code, only one out of three nitrogenous base, only one base is replaced by another base. Okay, for every amino acid, this triplet code is fixed. But when it is replaced by another amino acid, that means that amino acid will not form. Here now the new amino acid will be forming. Okay, new amino acid will be forming. This may produce another amino acid when this triplet coat is replaced instead of the normal which was already there in the sequence. Okay, already it was there in the sequence. This may produce in messenger RNA triplet code. Say for example, if from DNA this codes were copied by the messenger RNA when it was forming. Okay, and then this messenger RNA, instead of the code GAG, here the A and T will be replaced, so it will be code will be GUG. Now this triplet code GAG is responsible for the formation of amino acid called as glutamic acid. But due to the substitution of or replacement, okay, of the A by U here, okay, as we will see in the next diagram, then the triplet code has become GUG, and this is something new. Okay, and this will not produce glutamic acid. Since the code has changed, it is going to produce another amino acid called as valine. Okay, just see that just by change in one single nitrogenous base, amino acid becomes different. Okay, amino acid, and since amino acids are arranged in a sequence in a polypeptide chain, if even a single amino acid is different, then whole polypeptide chain becomes corrupt. It is not going to have the same function because polypeptide chains ultimately condense to form the protein. Okay, so the protein will become defective, as we will see here just now. Let us see here. This is the, I mean to say, chain of the sickle cell anemia. Okay, I told you again about this. Okay, once again I am repeating it here. Now here you see first on the top of the picture, I will show you by this. uh, See, you see, this is on the top of the picture. You are seeing the DNA, double strand of DNA, and just concentrate on this 3 days to the 5 days chain here. And if you concentrate on the sixth position, here the base is nitrogenous base or CTC. Okay, and in normal condition, against this CTC, messenger RNA which is produced, which brings the code from nucleus to cytoplasm to get attached onto the ribosomes and rough endoplasmic reticulum in production of the protein, that is GAG. So the code on messenger RNA is GAG, and this is normal, and this is responsible for production of the amino acid called as the glutamic acid. But if suppose change has taken place here, that means in a DNA where instead of the nitrogenous base pair TA, this is then replaced by AT. So in the DNA itself, the change has taken place. Instead of TA on the sixth position, this is now AT. That is, base pair has changed. Out of three base pair of a code, only one base pair has changed. That's why it is called as point mutation. Okay, because of this, now the messenger RNA which will form in the nucleus and which will come out in cytoplasm, it will have a different code. It will now have a GUG. Otherwise, what was the code? GAG. Now instead of here, it is the GUG. And as I told you in the last slide, instead of producing the glutamic acid, it will now produce the valine. And because of change in this amino acid, okay, in the sequence of this polypeptide chain, whole of the hemoglobin in the RBC will become defective. And because of this defective hemoglobin in the RBC, there will be condition will result what is called as sickle cell anemia. It is called as sickle cell, and you know about the sickle cell. I have told you in the clinical feature and other great details of the sickle cell. This will lead to lot of problems. Okay, moving to the next. So we are on the substitution mutation. Now we come to the second mutation, which is called as the frame shift mutation. Okay, now here the there may be a deletion or there may be another addition, and because of this deletion and addition, okay, here not the substitution is not taking place, okay, where one thing is replaced and another thing was put in. Okay, it is not like that. It is like that there. Here the normal sequence here in the code is CGT. It is the CGT which will code for an amino acid, and here because of the deletion of C nitrogenous base, okay, which was in normal sequence, what will happen? No substitution has, only the deletion has taken. That means nothing new has been added here as we have seen in the last example. But here because of this C is deleted, so that this whole code will shift, because two base pair cannot code for amino acid. It is needed that they should be in triplet. Okay, so this will shift further, and because of this, now instead of that, here it will be G, T and C. That means C of another sequence will be taken up to form the triplet code. So now the new code will be, because of deletion of C, the new code will become GTC. So this is GTC, and again if you see, this is the first base, this is second, and this will be third. So it is shifting has taken place throughout the downstream of this chain. Okay, so again GTC, which was otherwise CGT, now because here that shifting has taken place because of deletion of the one nitrogenous base pair, okay, nucleotide, whole nucleotide. Then if you see another condition in frame shift mutation, here instead of the deletion, it is after the insertion of a new base. Okay, here instead of C, now if C is added here, so instead of T here, after CG a new base C came here, so T was shifted to the next code. Okay, so this T which was in normal has been shifted to the new code, and thus this whole sequence downstream in this chain will be, they will have altered code, they will have altered, and this is said to be the frame shift mutation. So you got at the molecular level in a gene there may be two different types of mutation. What is called, first was called as substitution mutation and second is called as frame shift, and frame shift is more dangerous because everything after this addition or deletion will change. Okay, will change. So this were the molecular, I mean to say, level of the mutation, where we have explained you that how the mutation takes place. Point mutation, okay, point mutation or gene mutation takes place into the sequence of the triplet code. Okay, moving to the last slide. Now here, uh, why mutation occurs? Okay, you have understood the mutations are occurring, but you understand also why these changes are taking place. Okay, now first is that spontaneous, we don't know why these are taking place. Okay, we have yet not understood. So where no cause is known is called as the spontaneous mutation, right? But where the cause is known to us, okay, because of various research we know that this mutation may take place also because of this particular reason. Now, due to the effect of the environmental agents, okay, and these environmental agents which are responsible for mutation, that's why they are called as mutagens. They are called as mutagens because they are responsible for the mutations. And there are two classes of mutagens are known, that is chemical and physical mutagens. Okay, okay, chemical and physical mutagens. Let us come first to which are chemicals known for causing this mutation at the molecular level in DNA strand. Okay, now the best known example is the mustard gas. Okay, formaldehyde, which we inject in the dead bodies, formaldehyde, benzene, thalidomide, sorry, and then LSD, the drug. Okay, habit forming drug, LSD. Then physical agent, these were chemicals. Now physical agent is the high temperature, okay, is known physical mutagen. It is known physical when a person is constantly working in high temperature against the fireplace or something. Okay, that may be also a reason for this mutation. Okay, okay, that mutation may take place in somatic cell or it may take place in the germ cell. Okay, it may be in ovum or it may be in sperm. Okay, when they are forming. Okay, where meiotic cell division is taking place in ovary or in the say testes at that time because it is constantly going on. Okay, so this may take place at the germinal mutation at in the gametes. Okay, or it may take place in somatic, billions of somatic cells are there where they are constantly dividing, and at that time of the cell division this may take place. Now another cause where we know the effect, I mean say agent, okay, that is the mutagens. Well known mutagens are radiation. They are known since very long. Since very long we know about them, that the radiation, especially the natural ionizing radiation, and we are not aware about them, and they are since present in the soil, okay, or earth. They cause the natural ionizing radiation like sunlight. Okay, sunlight which contain ultraviolet rays, okay, or ultraviolet lamp itself. okay, they may cause this ionizing radiation, or that may change the sequence in the DNA molecules, okay, sequence. Now the radioactive thorium, they may also cause ionizing radiation. Radium itself, which is very well known, and the uranium, which are present in the earth, they are the source for this ionizing radiation, and thus they are known to cause the mutation in the genes, okay, in point mutation in the genes. Then there are artificial ionizing radiations are there, man-made, okay, man, they are not natural made. For example, X-rays, okay, X-rays are very well known, okay, that's why one should not go into the room where the X-rays are being taken or without protection, okay. Gamma rays, neutrons, etc. They are all very well known physical agents, radiations which are known to cause the ionizing radiation, and they are capable not only for the point mutation or substitution mutation, okay, or frame shift mutation, okay, they may also break the chromosomes or chromatids, okay, because they are quite fragile. As I said you when I was teaching you the chromosome, they are quite fragile and they can be broken, okay, with this ionizing radiation. This will lead to deletion or various kind of the structural abnormality in the chromosome or chromatid, okay. See, this may also in the polynucleotide strand level it may also call, okay, and not only at chromosome but at the DNA strand level, okay, DNA strand level or messenger RNA level, okay, any RNA or DNA level it may cause, okay. This completes the lecture on the mutations, okay. Hope you have understood it, okay. Thank you for watching this video, okay.